Powering human society with fossil fuels continues to release carbon dioxide into the atmosphere. Because carbon dioxide is a greenhouse gas, it will contribute up to 6.4 degrees of global warming by the year 2100. This unprecedented rate of change will increase aridity in our deserts, and ultimately the absence of water in these regions will result in the localized extinction of vegetation that provides key habitats for many woodland species across multiple taxa. The arid zone represents Australia's largest ecosystem, and it isn't as desolate as people believe. A complex mosaic of vegetation persists here, which supports many invertebrate species, amphibians, iconic mammals, birds, and more species of reptiles than anywhere else on the planet. Approximately 90% of these are actually endemic to Australia. In the last few decades, climate change impacts to species distributions have been assessed with species distribution models, which starts with a region of interest. In my case, this was Australia. We then divide the map into individual grid cells, and in each of these gridded cells, we insert temperature and precipitation values. Once we have completed this, we can then overlay our known occurrence records for our species and correlate these with the environmental conditions in that cell. From this, we can predict the current distribution. Once we have our predicted distribution, it's then a simple matter of changing environmental conditions, maybe increasing temperatures, lowering rainfall, and from this we can predict our future distribution. This allows us to know any range shifts, range contractions, or range expansions. Under global climate change, the problem with this technology though is that it tells us absolutely nothing about whether a species is capable of moving from its present distribution to its future distribution in the timescales imposed by rapid climate change. But this is important to understand for conservation because we need to know whether species can disperse quick enough and survive or whether they will die. So we drove across 1 million square kilometres throughout the central and eastern Australian deserts. This allowed us to obtain a large number of tissue samples from multiple arid zone landscapes with differing environmental features across a representative distribution of the species in question. This ensured a reliable estimate of annual dispersal rates could be calculated using genetic methods. Searching the potential retreat sites for small geckos, we were able to hand catch 874 individual tree detellers to obtain tissue samples for genetic analysis. With each of the 874 tissue samples, we performed a DNA extraction and then amplified a mitochondrial DNA gene to determine population structure. We also amplified 16 non-coding genes to obtain a genetic estimate of average annual dispersal using standard analysis techniques. To combine our genetic estimate of dispersal with our species distribution modelling results, we have designed a computer programming script. This starts with the predicted species distributions from 2010 and 2070 using a realistic climate scenario. We then dilated the present distribution with the mean annual dispersal distance multiplied by the number of years between the two predictions. In our case, this was 60 years. This allowed us to identify and quantify the areas in the future distribution, represented here in yellow, that can potentially be colonised within 60 years. We then dilated this yellow area with the mean annual dispersal distance multiplied by the number of years between the two predictions. Again, this was 60 years. This allowed us to identify and quantify the areas in the current distribution that can potentially reach the future distribution within the timescales of near future rapid climate change, here represented in purple. This century, rapid climate change will have a profound effect on global biodiversity, and species survival will be contingent upon their ability to disperse. Here I was able to combine species distribution modelling with a genetic estimate of dispersal and quantify that up to 41% of the tree deteller distribution is unlikely to reach regions of favourable conditions in the near future. This is really important information to establish for conservation because it lets us know which areas of the current distribution may require human intervention. We may need to facilitate dispersal. And secondly, it tells us which areas of land we should be conserving in our national reserve system so they will benefit species in the future. Additionally, this is a method that can be adopted wherever occurrence records and molecular data sets exist so that we can make rapid conservation assessments for species globally. Ultimately, these are reactive strategies to a global issue. In the longer term, we need to remove our dependence on fossil fuels and become a society committed to sustainable energy sources.